natural way developed technologies were to add greater comfort of human as value of human like the whole approach to understanding the natural phenomena become anthropocentric. Physics and chemistry gave rise to engineering, technology, and instruments which all work human comfort and welfare. The major utility of the biological world is the source of food. Biotechnology is the 20th century offshoot. Food and change our daily life as both quality to our health and food and production principle underlying biotechnology possess some application and highlighted and discussion in this unit. Herbert Boyer was born in 1936, brought up in a corner of Western Pennsylvania, where railroads and mines were destined the most young men. He completed graduate work at the University of the Pittsburgh. In 1963, allowed by 3 of post-graduate at Yale. In 1963, Boyer took assistant professor at University of California. By 1969, he performed study of a couple of restrictions in that of the E. coli bacterium with especially useful of properties. Boyer observed that this enzyme have the capability of cutting DNA strand into a particular fashion which left and has become known as stegians. This script and made pasting together piece of DNA a precise exercise. The discovery in turn led to a rich and rewarding conversation of Hawaii with a Stanford scientist named Stanley Cohen. Cohen has been studying small ringlets of DNA called plasmid and which float about freely in cytoplasm of a certain bacteria cell and replicate independently from the coding strand of DNA. Cohen had developed a method of removing this plasmid from the cells and then reinserting them in other cells. Combining this process is known as DNA splicing, enabled Boyer and Cohen to recombine segment of DNA in desired configuration and insert the DNA in bacterial cells, which could then act as manufacturing plant for a species of plant. This breakthrough on the basis upon which the discipline of biotechnology was founded. Biotechnology deals with the technique of using live enzyme from organisms to produce and process of useful of human. In this sense, making curd, bread, or wine, which all microbes in process could also be taught as a form of biology. However, it is used in a recipe today to refer to such of those processes which use genetically modified organisms to achieve the same on the large scale. For further, many other process techniques are also included in biotechnology. For example, in vitro fertilizing, leading to a test tube baby, synthesizing of a gene and using it, developing DNA sequence, or correcting a defective gene, all part of biotechnology. The European Federation of Biotechnology (EFB) has a definition of biotechnology that it encompasses both traditional view and modern molecular biotechnology. The definition by EFB is as follows: the integration of natural science and organism cells parts, therefore the molecular analogs for product and service. Principle of biotechnology. Among many, the two core techniques that enable birth of modern biotechnology are genetic engineering, technique to alter chemistry of genetic material, alter genetic material. Genetic engineering is a technique to alter the chemistry of genetic material DNA and RNA to introduce this into the host organism and thus change the phenotype of the host organism by a process engineering. Maintenance of sterile microbial contamination combines in a chemical engineering process to enable growth of only desired microbial eukaryotes in a large quantity for the manufacture of biotechnological products like antibiotic, vaccine, etc. Let us now understand the conceptual development of the principle of genetic engineering. You probably appreciate the advantage of sexual reproduction over asexual reproduction. The former provides opportunity of a variation and formulation of unique combination of genetic setup, some of which may be beneficial to organism as well as population. Asexual reproduction preserves the genetic information while sexual reproduction permits variation. The traditional hybridization process used in plants and animal breeding very often lead to the inclusion and multiplication of undesirable genes along the desired genes. The technique of genetic engineering, which includes the creation of recombinant DNA and use of a gene cloning or gene transfer to overcome this limitation, allows to isolate and introduce only one or set of a desired genes without introducing the undesirable gene into the target organism. Do you know the likely fate of a piece of DNA is somehow transferred to an alien system? Most likely, this DNA will not be able to multiply itself in its progeny of the organism, but when it gets integrated into the genome of the recipient, it may multiply and may be inherited along with the DNA along with the host of DNA. This is because the alien species of DNA has become part of a chromosome. This is because alien piece of DNA has become parts of a chromosome, which has the ability to replicate. In a chromosome, there is a specific DNA called origin of replication. In a chromosome, there is a specific DNA sequence called origin of replication, which is responsible for initiating replication. In chromosome, there is a specific DNA sequence called origin of replication, which is responsible for initiating replication. DNA sequence ORI, origin of replication, which is responsible for initiating replication. Therefore, for multiplication of any alien piece of DNA in an organism, it needs to be a part of a chromosome which has a specific origin of replication. Thus, an alien DNA is linked with the ORI is so that this alien species DNA can replicate and multiply itself into host. This can also be called as cloning or making multiple identical copies of any template. Let us now focus on the first instance of the construction of an artificial recombinant DNA molecule. The construction of a first recombinant DNA emerged from the possibility of linking gene encoding antibiotic resistant with a native plasmid autonomously replicating circular extra chromosome DNA of Salmonella typhi. Stanley Cohen and Herbert Boyer accomplished this in 1972. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating antibiotic resistant gene by cutting it out of a piece of DNA from plasmid, which was responsible for conferring antibiotic resistance. So the construction of first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with the native plasmid autonomous replicating circular extra chromosomal DNA of Salmonella typhi. Stanley Cohen and Herbert is this in 1972 by isolating antibiotic resistance by cutting a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. The cutting of DNA of a specific reaction became possible with the discovery of so-called oligonucleotide that is resistant enzyme. The cut off piece of DNA was then linked with the plasmid DNA. This plasmid DNA is a vector to transfer the piece of DNA attached to it. You probably know that mosquito acts as an insect vector to transfer malaria parasite into the human body. Mosquito acts as an insect vector to transfer the malaria parasite into human body. In the same way, a plasmid can be used to vector deliver to RNA species of a DNA into the host organism. This linking of antibiotic resistance, the linking of antibiotic resistance with the plasmid vector become possible with the enzyme DNA ligase, which act as a with DNA ligase, which act on cut DNA molecule and join there, and this makes a new combination of circular autonomously replicating DNA, circular autonomously replicating DNA. Created in vitro and is known as recombinant DNA. Created in vitro and is known as recombinant DNA. When this DNA is transferred into E. coli, a bacterium closely related to Salmonella, E. coli, a bacteria closely related to Salmonella, it could replicate using the new DNA polymerase enzyme and make multiple copies. The average of antibiotic resistance gene in E. coli was called cloning of antibiotic resistant gene in E. coli. The ability to copy of antibiotic resistant gene in E. coli was called cloning of antibiotic resistant gene in E. coli. You could have interpreted that there are three basic steps in genetically modified organism: identification of DNA with desirable, identification of DNA with desirable, introduction of the identification DNA into the host, introduction of the identified DNA into the host, maintenance of introduction of DNA into the host, and transfer of the DNA to its progeny. Maintenance of introduction DNA into the host and transfer of DNA to its progeny. Tools of recombinant DNA. Now we know that for foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have a key tools, that is example restriction and polymerase ligase vector and a host organism. Let us try to understand some of this in detail. In the year, the two enzymes responsible for restriction of the growth and bacteria. In the year 1963, two enzymes responsible for restricting the growth of a bacteria in E. coli were isolated. One of these added methyl group of DNA, and while other cut DNA. The latter was called restriction endonuclease. The first restriction endonuclease in 2 was function dependent on a specific DNA nucleotide sequence. Functioning dependent on a specific DNA nucleotide sequence was isolated and characterized 5 years later. It was found that hint to always cut DNA at molecule at a particular by recognized of sequence base pair hint2. This specific base sequence is known as recognition sequence for hint2. Besides hint2, today we know more than 900 restriction enzymes that have been isolated from 230 of a bacteria, each of different recognized sequence. The convention for naming this enzyme in the first letter of the name come from genus and second letter come from species. Genus, species, phyla, genus, species of the prokaryotic cells from which they were isolated. Example: E. coli comes from Escherichia coli RY13. In E. coli, R later derived from name of strain. R is strain. Roman number following the names indicate the order in which the enzyme were isolated from the strain of a bacteria. Restriction enzyme belong to a larger class of enzyme called nuclease. Restriction enzyme belong to restriction enzyme belong to larger restriction belong to a larger class of enzyme. Restriction belong to a larger class of enzyme called nuclease. There are two kinds of exonuclease and endonuclease. Exonuclease remove nucleons from the end of a DNA, whereas endonuclease cuts DNA at a specific position in the DNA. Each restriction endonuclease functions by inspecting the length of each restriction endonuclease functions by inspecting the length of a DNA sequence. Once it finds its specific segment, it will bind to the DNA and cut each two strands of the double DNA at a specific point in the sugar phosphate backbone. Example: each restriction endonuclease recognizes a specific palindrome in DNA. Each restriction endonuclease recognizes a specific palindromic sequence in DNA. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in DNA. Action of restriction enzyme. The enzyme cut both DNA strand at the same site. E. coli. The enzyme cut both DNA strand at the same site. E. coli cut the DNA between the bases of G and A, only when the sequence GATAC is present in the DNA sequence. GATAC is present in the DNA sequence. बोथ DNA स्ट्रैंड सेम साइड में कट करता है. E. coli स्टिकी एंड करता है और ब्लंट एंड करता है. E. coli स्टिकी recombinant DNA. Stemming information of recombinant DNA by action of endonuclease enzyme E. coli. Do you know the what palindrome? These are the group of letters that same words and repeat both for Malayalam as against word palindrome, where the same word in direction. Palindrome in DNA is a sequence of a base pair that reads same on two strands when orientation reading is kept the same. For example, the following sequence read the same on two strands, 5 3 direction. This is also true if we read 3 5 direction. Gatak 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 for E. coli. Restriction enzyme restriction enzyme cut strand of DNA a little away from the center of the palindromic side, but between the same two bases on the opposite side. Between two same bases on the opposite, little away from the center of the palindromic side, the center little away, but same bases के बीच में. Let's leave a single stranded portion at end. At end, ये क्या था single stranded portion. This leaves single stranded portion at the end. There are overhanging stretches called sticky end on each strand. Sticky end on each strand. These are named so because they form hydrogen bond. These are named because they form hydrogen bond with their complementary counterparts. This stickiness of end facilitates an action of a DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources of genome. When cut by the same restriction enzyme, the resulting DNA fragments have the same kinds of sticky ends, and this can be joined together end to end using DNA ligase. When cut by same restriction enzyme, the resulting DNA fragments have the same kinds of sticky ends, and this can be joined together end to end using DNA ligase. When cut by same restriction enzyme, the resulting DNA fragment have the same kind of sticky end, and this can be joined together end to end using DNA ligase. Foreign DNA, vector DNA, plasmid, foreign DNA, same restriction enzyme cutting both foreign DNA and vector DNA at a specific location. Where DNA ligases join foreign DNA to plasmid, recombinant DNA molecule transform cells divides E. coli colony or you may have realized that normally, unless one cut a vector and the source of DNA with the same restriction, then the recombinant vector molecule cannot be created. Unless one cut the vector and source of DNA with the same restriction, the recombinant vector molecule cannot be created. Separation and isolation of DNA. The cutting of a DNA by restriction and the nucleus results in the fragmentation of DNA. The cutting of a DNA, cutting of a DNA is a cutting by restriction and the nucleus results in the fragment of DNA. This fragment can be separated by a technique known as gel electrophoresis. Fragment separate by gel electrophoresis. Since DNA fragments are negatively charged molecules that can be separated by forcing them towards the anode under the electric field through a medium through a medium or matrix. Nowadays, the most commonly used matrix is agarose gel, which is a natural polymer and extracted from a seaweed. Extracted from a seaweed. Seaweed is a red algae. A natural polymer extracted from a seaweed. The DNA fragments separate resolve according to their size. Through sieving effect. Provided by the agarose gel. Hence, smaller the size, further it moves. Smaller the size, further it moves. The separated DNA fragment can be visualized only after staining the DNA with a compound known as ethylium bromide followed by exposure to UV radiation. You cannot see pure DNA fragment in the visible light without staining. You cannot see pure DNA fragment in the visible light and without staining. You can see bright orange colored bands of the DNA in ethylium bromide. Bright orange colored band of DNA in ethylium bromide strength gel exposed to UV light. The separated bands of a DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elution. Extraction को बोलेंगे extracted को agarose the separated bands of DNA are cut out from agarose gel and extracted from the gel piece. This step is known as elution. The DNA fragment purified in this way are used for constructing recombinant DNA by joining them with cloning vector. The DNA fragment purified in this way are used for constructing recombinant DNA by joining with cloning vector. You know that the plasmid and bacteriophage have the ability. Plasmid and bacteriophage have ability to replicate within the cell independent of the control of a chromosomal DNA. Bacteriophage is because of their high number per cell have very high copy of number of their genome within the bacterial cell. Some plasmids may have only 1 or 2 copies per cell, whereas the other may have 1500 copies per cell. Some plasmid may have only 1 to 2 copies per cell, whereas other may have 15 say 100 copies per cell. The number can go even higher. The number If we are able to link elimination of DNA with the bacterial or plasmid, we can multiply it a number equal to a copy of number of a plasmid or bacterial. Vector used at present are engineered in a such way that they have the linking of foreign DNA and selection of recombinant from non-recombinant. The following features that are required to facilitate turning into a vector. ORI, origin of replication. This is a sequence from where replication starts. ORI replication starts. Any piece of DNA if link this sequence can be made to replicate within the host cell. This sequence is also responsible for controlling the copy number. Start करता है ORI. The controlling center करता है ORI number control करता है of a linked DNA, and so it once recover many copies of a DNA, it should be cloned with into a vector or it can support his high copy number. Selectable marker. In addition to ORI, the vector requires a selectable marker which help in identifying and eliminating non-transformant, and selectively permitting the growth of a transformant. Alright, the vector requires a selectable marker which help in identifying and eliminating non-transformant and selectively permitting the growth of transformant. Transformation is a procedure. Transformation is a procedure through which piece of DNA is introduced into a transformation is a procedure through which piece of DNA is induced into the host of a bacterium. Transformation is a procedure through which piece of DNA is introduced into the host bacterium. Process of you will study in supplementation. Normally, genes encoding resistance to antibiotics such as ampicillin, chloramphenicillin, tetlacin, or kanamycin, etc., are considered a useful selectable marker for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. Transformation is a procedure through which piece of DNA is introduced in a host bacterium. Normally, the genes encoding resistance of antibiotics such as ampicillin, chloramphenicillin, tetracycline, or kanamycin, etc., are considered useful selective marker for E. coli. The normal E. coli cells do not carry resistance against any of these antibodies. The normal E. coli cells do not carry any resistance against any of this cloning site. In order to link alien DNA, the vector needs to have a very few, preferably single recognized cells for the commonly very few होनी चाहिए, preferably single recognized cells for the commonly used restriction. Single recognition cells होनी चाहिए for the commonly used restriction. Presence of more than one recognized cells within the vector will generate several fragments which will complicate the gene cloning. The ligation of alien DNA is carried out. The ligation of alien DNA is carried out. Ligation of alien DNA is carried out at a restriction site. Ligation of alien DNA is carried out a restriction site present in one of two antibody resistant genes. For example, you can ligate foreign DNA at BAM-HI sites of tetracycline genes which present in a vector PBR322. Presence of more than one recognized sites within the vector will generate cells which complement the ligation of alien DNA is carried out at a restriction site present one of the two antibiotic resistant genes. One of the two antibiotic resistant genes. For example, you ligate foreign DNA at BAM-H1 site of tetracycline, BAM-H1 site of tetracycline, BAM-H1 site of tetracycline. Regions of vector in PBR2. डायग्राम बहुत इंपॉर्टेंट है. आगे से कमेंगे PBU2, 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 2 ROP से कनेक्ट है. टर्शियरी में करेंगे BAM-H1 साल H1, BAM-H1 साल H1, BAM-H1 साल H1. एम्पिसिलिन में आता है P, तो PST1 PBU1, PST1 PU1, PBU1, PST1 PBU1. टेट्रासिलिन BAM-H1, टेट्रासिलिन BAM-H1 साल H1, BAM साल 1, BAM साल और इकोरा क्लाव1 हिम3, क्लाव1 हिम3 इकोरा प्रेजेंस में. PBU और में ROP में क्या होता है PBU. Equal connected PBU showing restriction sites in HIM3 equal 1 BAMH SAL PBU PH class ORI and antibiotic resistance ampicillin and tetracycline. ROP codes for the protein involved in the replication of ROP codes for protein involved in the replication of plasmid. The recombinant plasmid will lose tetracycline resistance due to the insertion of a foreign DNA, but can still be selected out from non-recombinant ones by plating the transformants on the tetracycline containing medium. The transformant growing on an ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinant will grow in ampicillin containing medium but not on containing tetracycline. But non-recombinant will grow on the medium containing both. On the antibody, in this case, one antibiotic resistance help in selecting the transformant, whereas the other antibiotic resistance get inactivated due to the insertion of alien DNA and help in selection of recombinants. Selection of recombinant due to the inactivation of antibody is a cumbersome procedure because selection of recombinant due to inactivation, selection of recombinant due to insertion due to inactivation of antibiotic is cumbersome procedure because it requires simultaneous plating of two plates having different antibiotics. Therefore, alternative selective markers have been developed which differentiate recombinant from non-recombinant on the basis of their ability to produce color in the presence of chromogenic substance. In this, recombinant DNA is inserted within the coding of sequence of the enzyme beta-galactosidase. This results into inactivation of the gain of synthesis of this enzyme, which is referred as insertion inactivation. The presence of a chromogenic substance gives blue. Presence of chromogenic substance gives blue-colored colonies if the plasmids of the bacteria does not have an insert. Presence of insert result into the insertion inactivation of the beta globin gene, and colonies do not produce any color. These are identified as the recombinant colonies. Recombinant colonies. The presence of chromogenic substance gives blue colors. Presence of chromogenic substance gives blue color colonies if plasmid of the bacteria does not have an insert. Insert नहीं हो रहा है ना तो presence of insert result into insertion inactivation of the beta glycan and colonies do not produce any color. These are identified as recombinant colonies. Vector of cloning genes in plants and animals. Have been vector surprise and had learned a lesson transforming genes into a plants and animals from bacteria and viruses which have no only stages. How to deliver genes on a transfer you yourself of course them to do what bacteria or virus want. For example, Agrobacterium tumefaciens, a pathogen or dipole plant, Agrobacterium tumefaciens, pathogen for dipole plant is able to deliver a piece of DNA, tDNA, piece of DNA, tDNA, transfer normal plant cell into a tumor, transfer tDNA to a normal plant cell to tumor cell करेगा and direct and this tumor cells produce the chemical required by the pathogen. Similarly, retrovirus in animal have the transform a normal cell to the cancer cell, a better understanding of the art of delivering genes of pathogen in the better their eukaryotic host had generated knowledge to transform these tools of pathogen into useful vector for traveling genes of interest to human. The tumor TI plasmid of Agrobacterium tumefaciens has now been modified into cloning vector, which is no more pathogen to the plant, but is still able to use mechanism to deliver the genes of into varieties. Similarly, retroviruses have also disarmed and are now used to deliver desired gene. So once a gene or DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial cell or animal where it's multiplied. Competent host offer transformation with recombinant DNA. Since DNA is hydrophilic nature, molecule it cannot pass through a cell membrane. Why? In order to force bacteria to take up plasmid, the bacteria cell must be made up of components to take up DNA. This done by treating a specific concentration for divalent cation such as calcium, which increase the efficiency with DNA, which increases the efficiency with which DNA enters. With which DNA enters the bacterium through pores in its cell wall, which increases the efficiency of DNA. Because this is done by treating them with specific concentration of and which increases the efficiency with which DNA enters the bacterium through pores in the cell wall. Recombinant DNA molecule can either be forced into a cell by incubating the cells within the recombinant DNA on ice and followed by placing them briefly at 42 degrees Celsius heat shock and then putting them back on the ice. This enables the bacteria to take up the recombinant DNA. This is not only way to introduce alien DNA into the host. A method known as microinjection. Recombinant DNA is directly injected in the nucleus of an animal. Nucleus and another method suitable for plant. Another method suitable for plant cells are recombinant with highly velocity microparticles called tungsten. In another method suitable for plant. Cells are bombarded with high velocity microparticles or gold or tungsten microparticles of gold or tungsten coated with DNA in a method known as biologistic or gene gun. And last method is uses disarm pathogen vectors which when allowed to infect the cell transfer the recombinant DNA into the host. Now that we have learned about the tools of recombinant DNA, let us discuss the process of facilitating recombinant DNA. The process of recombinant DNA technology involves several such as isolation of a DNA, fragmentation of a DNA, biorestriction endonuclease, isolation of a desired DNA fragment, ligation of a DNA into the vector, transferring the recombinant DNA to the host, culturing the host cell in the medium at a large scale, and extraction of a desired protein, isolation of genetic material DNA. Recall that nucleic acid is genetic material of all organisms without exception. In majority of organisms, this is a dioxy DNA. In order to cut DNA with a restriction enzyme, it needs to be in pure form, free form of other macromolecules. Since the DNA is enclosed within a membrane, and we have to break cell open to the release the DNA along with the other macromolecules such as RNA, protein, and polysaccharide isolipids. Other macromolecules such as RNA, protein, polysaccharide, and isolipids. This can achieve by treating in bacterial cell, plant cell, or animal tissue with enzymes such as lysozyme, bacteria, cellulose, plant cell, skinless fungus. You know, genes are located on long molecules of DNA. Genes are located on long molecules of DNA, intertwined such as histone, intertwined with the protein such as histone. The RNA can be removed by treating the ribonuclease, whereas RNA can be removed by treating the ribonuclease, whereas the protein can be removed by treatment with protease. Other molecules can be removed by appropriate treatment and purified DNA ultimately precipitate out the addition of ethyl ethanol. This can be seen in the collection of a fine thread of suspension. Cutting out DNA as a specific location. Restriction enzyme digestion are performed by incubating purifying DNA molecule. Also, with the restriction enzyme at the optimal conditions for the heat-specific enzyme, alveolar gel electrophoresis is employed to check the progression of a restriction enzyme digestion. Alveolar gel electrophoresis is employed to check the progression of restriction enzyme digestion. DNA using electric terminal and it moves towards the positive electron, and this process is repeated with the vector DNA also. The joining DNA involves viral process. The joining DNA involves a viral step after having cut a source DNA as wall as a vector DNA and with a specific restriction enzyme. Joining of a DNA involves viral process. Joining of DNA involves viral process. Joining of DNA after having cut off a source DNA as well as the vector DNA with a specific restriction enzyme. The cut out gene of and from the source of DNA and cut factor with the specific are mixed and ligase is added. Here the cut out gene of interest from the source of DNA and cut the vector with a space mixed ligase is added. This results in the preparation of recombinant. This results in the preparation of recombinant. 9333. Application of gene of interest using PCR. PCR stands for polymerase chain reaction. Polymerase chain reaction. Amplification of gene of amplification of gene of interest. Amplification of gene of interest. PCR stands for polymerase chain reaction. PCR polymerase chain reaction. Amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. And multiple copies of gene or DNA of interest is synthesized in vitro using two पहला डीनेचुरेशन करते हैं, फिर दूसरा एनिलिंग करते हैं, फिर तीसरा एक्सटेंशन करते हैं 1 बिलियन एम्पलीफायर 30 डीएनए पॉलीमरेज टैग पॉलीमरेज डीऑक्सी टैग पॉलीमरेज प्लस डीऑक्सी न्यूक्लियोटाइड्स प्राइमर्स डबल स्ट्रैंड डीएनए प्राइमर 30 सेकंड इस तरह से सिस्टम डीनेचुरेशन प्राइमर एनिलिंग एंड एक्सटेंशन ऑफ प्राइमर. Using two sets of primer, small chemically synthesized oligonucleotides that are complementary to the region of a DNA. DNA with in multiple of copies of gene or DNA or interest in synthesis in vitro using two sets of primer, small chemically synthesized oligonucleotides that are complementary to the region of DNA and the enzyme DNA polymerase. The enzyme extend the primer using the nucleotides provide a reaction and genomic DNA as a template. Provide the enzyme extend the primers using the nucleotides provided the reaction and the genomic DNA as a template. If the process of replication of DNA is replicated many times, the segment of DNA can be amplified approximately 1 billion times. 1 billion copies are made. Such repeated amplification is achieved by use of a thermostable DNA polymerase isolated from bacterium Thermus aquaticus, which remain active during high temperature and uses denaturation of double-stranded DNA. The amplified fragment, if desired, can now be used to ligate a vector to a further cloning. Insertion of recombinant into the host cell environment. There are several methods to introduce the ligated DNA into the recipient. There are several methods to introduce ligated DNA into the recipient. There are several methods to introduce ligated DNA into the recipient. There are several methods of introducing the ligated DNA into the recipient cells. There are several methods introducing the ligated DNA into the recipient cells. Recipient cells, after making them a component to receive attack of the DNA present and surrounding. So, if recombinant DNA is bearing polarization to an antibiotic, for ampicillin is transferred into E. coli, the host cell becomes transformed into ampicillin-resistant cells. If we spread the transformed cell on agar plates containing ampicillin, only transformants will grow. Only the untransformed recipient will die. Since due to ampicillin resistant gene, one is able to select transformed cell in the presence of ampicillin. The ampicillin resistant gene in this is called as a selectable marker. Obtaining the foreign gene product. When you insert a piece of the alien DNA in a colonimeter and transfer it to a bacterial plant or cell, the alien DNA can multiply. Almost all recombinant technology. The ultimate aim to produce a reliable protein has this where need to recombinant DNA to be expressed. The foreign gene can get expressed under appropriate conditions. The expression of a foreign genes in host cells involve understanding of many technical details. After having cloned the gene of interest and having optimized conditions to induce the expression of the target protein, one has to consider the protein in a life. Can you think of any reason where any life production? If any protein encoding gene is expressed in heterologous host, it's called recombinant protein. If any protein encoding gene is expressed in heterologous host, if any protein encoding gene is expressed in heterologous host, it is called recombinant protein. Recombinant protein. The cells harboring clone gene of interest may be grown on a small scale in a laboratory. The culture may be used for excreting desired product and then providing by different separation taking the cells and multiply in continuous culture system where the use in a tray and drain from one side to fresh medium is added and from other to maintain their physiologically active log exponential rate. This type of culture method produces larger biomass leading to the high rates of a desired production. Small volume culture cannot yield appreciate quantities of product. To produce large quantities, the development of bioreactor where large volume of 100 to 1000 liters of culture can be processed was required. The bioreactor can be thought be as a vessel in a raw material of biological converted into a specific product. Individual enzymes are using microbial plant, animal or human, individual enzymes, etc. using microbial plants, animal or human cells. A bioreactor provides optimal conditions for achieving the desired product by providing optimum growth of temperature, pH, and substrate or solid, vitamin, etc. Oxygen, etc. The most commonly used bioreactors are stirring type, which are shown. Motor form, flat, culture, acid base and steam for sterilization, increase surface area for oxygen transfer. Sparged stir tank bioreactor, simple stir bioreactor. A stir tank reactor uh, is really cylindrical or cervical case facilitates mixing of a reaction content. The still facilitates even mixing of even mixing and oxygen availability throughout the bioreactor. The alternative air can be bubbled through the reactor. If you look the closely your bioreactor and aggregate system, the oxygen delivery system and form control system, a temperature control system and pH control system and sampling ports of the small volumes of the culture can be withdrawn critically. Downstream process after completion of a biointegration, the product can be subjected through a series of process before it's ready for marketing and finished. The process includes separation, purification, and which are regarded by the downstream process. The product has to be formulated with a suitable preservation. Such formulation has to undergo also clinical trial in case of drugs. Strict quality control testing for each product is also required. The downstream process and quality control testing vary from drug to biotechnology with the large scale production and marketing by product using living organisms cells in the medical field only. Chemistry of a DNA and construction of DNA. This key process called recombinant DNA technology. In this process involves precision and different DNA ligases appropriate viral accelerated ferry foreign DNA into the host acquisition of foreign DNA functional protein and finally making suitable formulation for marketing and large scale program involve uses of bioreactor.